Hi everyone, welcome to Jelus Microtech. Today we are going to know about structure of virus. Characteristics of virus. First of all, we know about what is virus. Viruses are the smallest infectious particle. Viruses are obligate intracellular parasite that depend on the biochemical machinery of the host cell for replication. They are extremely small in size and simpler than bacteria. They are ultramicroscopic, visible only in the electron microscope. They are dependent parasites that cannot survive on their own. They require living organisms to multiply as they cannot live on their own. Structurally, viral particles are simpler and minute in nature. A wide variety of shapes and sizes have been observed. Viruses can be measured in unit of nanometer. Father of virology is Martinus William Bainbridge. Martinus William Bainbridge called a filtered infectious substance as Contagium vivum fluidum, ranging in diameter from 18 nanometer to 1500 nanometer. Viruses typically contain either deoxyribonucleic acid as DNA or ribonucleic acid as RNA, but not both. Viruses require host machinery for replication, cannot synthesize enzymes or protein on their own. Viral components are assembled and do not replicate by division. Virus can infect all the life forms on earth, that is invertebrates, vertebrates, plants, protista, fungi and bacteria. Viruses have a naked capsid or envelope morphology. More than 2000 species of virus have been observed that infect animals and humans. Viral components are genetic material, nucleic acid, DNA or RNA containing viral genes, but not both, capsid, protein shells surrounding the genetic material, envelope, lipid bilayer derived from the host cell membrane. Proteins. Structural, non-structural and regulatory proteins are present in the viral compounds. Viral structure types. Helical, isohedral, complex. These are the viral structural types. Viral size typically ranges from 20 to 400 nanometer in diameter. Some virus like smallpox can be larger, up to 450 nanometer. These are the shapes of virus. Icosahedral structure. The protein subunits assemble into symmetric shell that converts the nucleic acid containing core. Icosahedral is a polygon with 20 vertices, 20 facets and 30 edges. Each facet is equilateral triangle. Helical structure. The protein subunits and nucleic acid are arranged in a helix or spiral tube-like structure. Capsules are coiled around the central axis to form the helical structure. For example, tobacco mosaic virus is a helical virus. Complex. Complex viruses are, for example, for complex viruses is bacteriophage. It infects bacteria. Structure of virus. There are two major structure of virus called naked nucleocapsid virus and enveloped virus. First, capsid. Capsid is outer shell that surrounds the viral nucleic acid, composed of capsomer subunits collectively protect the nucleic acid from the environment. Capsid has two functions: to protect the genome from external harmful factors or agents, or to introduce the viral genome into the host cell. Capsomers, individual proteins associated into progressively larger units. Envelope. Lipid and proteins, basically a modified version of outer membrane. Envelope spikes. bind to the cell surface protein. During the release of animal virus, a part of host membrane is taken. Nucleic acid. Virus contain either DNA or RNA, not both. Possess only the genes to invert and regulate the metabolic activity of the host cell. For example, hepatitis B, 4 genes, and herpes virus, 100 genes. No viral metabolic genes as the virus uses the host metabolic resource. Proteins. There are three types of proteins: structural protein, non-structural protein, regulatory protein. Structural protein, for example, capsid and envelope. Non-structural protein, replicase, transcriptase. Regulatory protein, for example, transcription factors. Functions: attachment and entry, replication and transcription, assembly and release for regulatory protein. Spike protein. The spike protein location is surface of the envelope. Its function is attachment to the host cell, fusion with the host membrane. Matrix protein. Location between envelope and capsid. Function: stabilize envelope and interacts with host cell. Nucleocapsid. Composition: protein RNA complex. Function: protects the genetic material and regulates replication. Tail fibers, end of the viral particle, attachment to the host cell, facilitate entry. Base plate, end of the viral particle, stabilize the tail fiber, facilitates attachment. Thank you. I hope we all know about structure of virus.